Hi, my name is Caitlin Moggle. I'm a research scientist at University of Washington, Tacoma, and I'm here to share with you the results of our HB 3114 funded project, where we built a biophysical model of Coos Bay to look at the interaction between eelgrass and water quality. Eelgrass, or Zostera marina, is the common type of seagrass in estuaries along the Pacific Northwest coast. Seagrasses are recognized for their many valuable services to estuarine ecosystems and to society. For this project, we were focused on the service at the bottom here because a growing number of studies have found that seagrasses can have local effects on carbonate chemistry and dissolved oxygen. However, the importance of seagrass mitigation of ocean acidification and hypoxia and the scales at which mitigation can be realized are still not fully understood. Interest in OAH mitigation strategies has grown rapidly because of the threat that OAH imposes on natural communities, including ones of importance to humans like salmon and shellfish. Just a quick refresher on how this mitigation hypothetically occurs. During photosynthesis, these plants take up carbon dioxide from the water column and release dissolved oxygen, resulting in increased pH and DO. During respiration at night, however, the opposite occurs. This results in a day-night or diel cycle, where pH and DO increase during the day and decrease at night. The thought is that inside of eelgrass beds, because of their high productivity, pH and DO will be much higher during the day, even though they decrease at night, and that overall, on average, pH and dissolved oxygen will be higher inside of eelgrass beds compared to outside of eelgrass beds. Because seagrasses are highly productive ecosystems, it's thought that they have large mitigation potential. There are various methods that have been used to try to measure or estimate OAH mitigation by seagrasses. Some of the ways that we've seen in the literature include simple box models, mesocosm studies, comparisons inside versus outside of beds, or comparisons across interannual variability. And finally, ecosystem models. These are highly quantitative models that describe both the biological, chemical, and physical factors occurring in estuaries. Given that it's critical to understand how mitigation by seagrass translates at estuary-wide scales under highly variable environmental conditions, it's likely that ecosystem models would be particularly useful. These models provide the opportunity to explore ideas, test hypotheses, and provide insights into ecosystem scale processes that are difficult to explore with real-world experiments. So our aim for this project was to produce a biophysical model of Coos Bay complete with eelgrass, and then use the model to understand dynamics of pH, dissolved oxygen, and eelgrass in the estuary, and ask questions about what conditions confer vulnerability to ocean acidification and hypoxia, and how eelgrass abundance alters pH and dissolved oxygen in the estuary. Recent declines in Coos Bay eelgrass, as shown in these pictures, further highlight the need to understand the consequences of variability in eelgrass abundance for OAH mitigation and other important ecosystem services. Coos Bay is home to ecologically, economically, and culturally important ecosystems and species that are vulnerable to ocean acidification and hypoxia. The long history of research and monitoring in this estuary, including the presence of South Slough National Estuarine Research Reserve, means that we have access to a wealth of data and information upon which to inform model development. Specifically, we were able to leverage an existing water quality and eelgrass monitoring data, a hydrodynamic circulation model of the estuary, and a biogeochemical model framework. Our three-dimensional Coos Bay model starts with the hydrodynamic model for the year 2014. The hydrodynamic model is based on the river inputs, which are the blue dots in the map, and an open ocean boundary. We initially run the hydrodynamic model and then use the hydrodynamic output to calculate the transport of water quality variables with the addition of sources and sinks and other biogeochemical processes. This schematic shows the variables and processes captured in the biogeochemical model. It simulates biogeochemical processes such as plankton productivity, nutrient uptake and release, settling and resuspension, and others, and ultimately can compute resulting dissolved oxygen and pH levels. As shown in the bottom right here, the eelgrass submodel is divided into two compartments, above and below ground, which each have their own kinetics that depend on light availability, nutrient uptake, and mortality. This type of model has been used extensively in the Salish Sea to study aspects of environmental change, including OAH, but also sea level rise and marine heat waves. The eelgrass submodel was implemented, tested, and validated in the Salish Sea. So we're building on lots of previous expertise in applying this type of model to Coos Bay. To inform development of the biogeochemical model, we gathered existing water quality and eelgrass data from various sources. The locations for existing water quality data are shown on the left, and on the right is in green is the maximum observed extent eelgrass layer from PMEP. And we also collected observed point data to refine eelgrass biomass within that extent. Together, the water quality and eelgrass data is used to parameterize the biogeochemical model, establish the initial conditions, and validate model outputs. Despite this being a data-rich estuary, we've still run into data limitations. Data available specifically for the year 2014 has been limited, and nutrient and carbon data is particularly limited, especially for freshwater end members. So we've had to apply either seasonal averages or rely on regional estimates or data from other nearby systems. So the first thing that we do is compare observations then at other stations throughout the estuary with the model's predicted outcomes. So here I'm showing model results in orange compared to observations in blue for dissolved oxygen, pH, nitrate, and ammonium. And this is at one particular station. This is at Velino Island in South Slough, but we looked at these figures for all of the water quality stations that you see on the map here on the right, and compared model results to observations through our calibration and validation process. We did a similar process for the eelgrass model, where the model results for above and below ground biomass are given in the lines here, and then observations for above and below ground biomass are given in the box and whisker plots. And we compared, there's lots of variation obviously in the observed data, even just at these two locations, but we compared the model outputs to the median values from the observations for eelgrass biomass, and saw relatively good performance of the eelgrass model to predict biomass, both above and below ground biomass. We used the model to run 3 scenarios for eelgrass abundance along a continuum from high eelgrass coverage to no eelgrass. On the right is our high eelgrass abundance scenario. We used the maximum observed extent coverage map from PMEP, which represents all the areas that had eelgrass in the 1978, 2005, and 2016 surveys. In the middle, the current extent is based on the 2016 survey, and then finally a no eelgrass scenario. So let's dig into some of the results. So here we're looking at time series of dissolved oxygen and pH across the 3 simulations or scenarios. The yellow line represents the no eelgrass scenario, the light blue is the current extent scenario, and dark blue is the maximum observed extent scenario. We're looking at results from within eelgrass beds at 3 different sites, moving from north to south, top to bottom. The first main takeaway is that there's an increase in variability of both dissolved oxygen and pH in the scenarios with eelgrass. In particular, for dissolved oxygen, we see higher highs and lower lows, but we don't necessarily see a change in the mean dissolved oxygen condition when we have eelgrass versus not. For pH, however, we definitely see higher highs, but we don't necessarily see lower lows. So in general, the average pH was increased by including eelgrass in the model. And these, like I said, these locations are within eelgrass beds. As you move further away from eelgrass beds in the model, you see a decreasing influence of eelgrass on dissolved oxygen and pH. Another way to look at the results is to integrate over space and time. So we calculated the exceedance of a biologically relevant dissolved oxygen threshold, that being below 6.5 milligrams per liter, which is based on salmon physiological requirements. We then calculated the change in exceedance of that threshold over space and time as you move between scenarios. So over the whole estuary, there is actually a slight increase in volume and time exceeding the threshold as you move from a no seagrass to a current extent scenario, and then another small increase as you move from current extent to maximum extent. But within the estuary, there were some places where DO conditions improved or became less harmful, those are the red areas, and some places where DO conditions became more harmful, those are the blue areas. These are the same types of figures, but showing exceedance of a biologically relevant pH threshold based on previous research showing that Olympia oysters perform best at pH greater than 7.8. Again, we calculated the volume and time exceeding that threshold and looked at the change between scenarios. So moving from no eelgrass to current extent, there was a 7.6% decrease in the volume and time exceeding that threshold, and then a further 5.3% decrease as you move from current to maximum observed extent of eelgrass. An improvement generally in the pH conditions across the estuary, but again, some areas had essentially no change, those are in the white and yellow, and other areas in green and blue where we saw larger improvements in pH conditions. We know that eelgrass provides many important ecosystem services, but if we were going to focus eelgrass conservation and restoration efforts based solely on the OAH mitigation service, the results of our study help us to identify several areas where we are likely to see benefits for dissolved oxygen and pH. I've circled 3 such areas where we saw improvements in both DO and pH conditions based on those biologically relevant thresholds. So the areas outside of Kentuck Inlet, Haynes Inlet and North Slough, and Airport Flat, for example. These areas appear to have the right conditions for OAH mitigation by eelgrass. But of course, there are many other factors that should be considered when planning eelgrass conservation and restoration. This is just one facet of those decisions. In conclusion, we successfully developed a biophysical model of Coos Bay that simulates water quality and eelgrass in the estuary. We found that simulation results in models with eelgrass more closely matched observations, especially the high variability of water quality parameters that's common in estuaries. Now that the model is built, it can be used in other research and management applications. For example, we can run additional scenarios around coastal development or climate change and look at the impacts on the Coos Bay estuary. Due to interacting biological and hydrological factors, the OAH mitigation by eelgrass in Coos Bay differed both spatially and temporally, and there were differences between dissolved oxygen and pH. For DO, we found that there wasn't a change in mean DO, but that there was higher variability, especially during summer when eelgrass are net productive. For pH, we did see an increase in the mean and also higher variability. Overall, we saw reductions in the exceedances of biologically relevant pH and carbonate mineral saturation state, or omega, thresholds, indicating that OAH conditions were improved by the presence of eelgrass in the model. On the other hand, there was a slight increase in the exceedance of the DO threshold. But again, there were spatial differences, so some locations improved while others were worse off. And we can use these fine-scale details to help plan eelgrass restoration and conservation efforts towards ecosystem benefits. With that, thank you so much to our funding from Oregon Ocean Science Trust and the OAH Council, and thank you for the folks who provided data that helped us inform model development and validation. Thanks for watching.